Penicillin is an antibody of penicillin, a fungi species which prevents gram-positive bacteria from forming cell walls. It has been an effective drug against bacterial infections since it targets bacteria-specific proteins and not human proteins. Penicillin only takes effect against gram-positive bacteria because gram-negative bacteria have a lipopolysaccharide in a protein layer which prevents the drug from penetrating the bacteria's cell wall. As bacteria divide, their cell walls open up holes, which are then backfilled with newly produced peptoglycogen and reconstructs the cell wall. Penicillin attacks bacterial cells by inhibiting specific proteins in its peptoglycogen, which are responsible for cell wall biosynthesis. Penicillin inhibits the bacteria from closing the holes in its cell walls. Without the cell wall, the bacteria do not have protection, are more susceptible to osmotic imbalance, osmotic lysis, and thus cell death. Since bacteria are prokaryotes, their cells are very much different from ours. Other pathogens like fungi and parasites are eukaryotes and have cells which are more similar to ours, making it harder to develop drugs against them. Penicillin, an antimicrobial drug, does not affect humans because we do not have cell walls with peptoglycogen. Thus, the drug will not be able to do its function inhibiting cell wall synthesis and cell lysis. Penicillin may act more effectively on one type of bacteria compared to other types. Sometimes some bacteria can develop a resistance against it. These penicillin-resistant bacteria are able to develop penicillinases, which can degrade the drug. They can transfer this ability to other bacteria through conjugation using a sex pili. In addition to this, some bacteria have the ability to alter their peptoglycogen layer, making it impossible for the drug to work properly.